work.
Okay, so the last lecture will be just the research presentation about one project, one of our last projects, which includes rare neurodegenerative disease, mitochondrial function, and drug development or drug target research, and a lot of imaging. So I'm going to specific details, even if they are maybe not so interesting. I think they are still valuable to understand what is required to be, how to say, sure about your conclusions and how difficult sometimes it is to do something. Some of the slides are you have seen already before in previous presentations with pictures. So one of the diseases we are studying is this Wolfram syndrome. It is a genetic disorder, and it is characterized by diabetes, and in addition to diabetes also optic atrophy, deafness, and brain atrophy. It is considered to be also neurodegenerative and neurodevelopmental disease. Neurodegenerative because the brain stem is degenerating, at least, and also maybe other areas as well. Diabetes in these patients could be managed more or less with current therapies, but the problem is that the neurological symptoms can't be managed. There is no drugs, and usually these are the main problems for patients. They will first get diabetes, this could be managed, but then the optic atrophy and deafness will start. They anymore can't see properly, can't hear properly. So, and this usually happens already when they are 10 years ago, sometimes maybe in 20s, but mostly when they are relatively young, and life expectancy is maybe to 30s, sometimes to 40s, and it's quite, how to say it simply, quite bad disease as other such kind of genetic neurodegenerative diseases. So there is no real treatment, and the reason why there is no treatment, as in case of other diseases, is that even if we know the original protein, we don't know exactly its function. If we don't know the function of that protein, then it's quite difficult to find some kind of targets also. It's not monogenic disease. There is two genes involved. One is the wolframin, which is giving the name to this disease. It is, you can see the multipass protein in endoplasmic reticulum membrane. It has big portion inside of the ER in lumen, and it has also big domain in cytosol. And it's big, 100 kilodaltons or so. And the mutations are in many different places. So you see these red dots, this is the protein, red dots are, how to say, mutations. So you have mutations in many places. And it's not that one mutation dominates, you have a lot of different mutations in patients also. This is making the gene therapy quite complicated, because you have your therapy against all these mutations. So at the moment it is not realistic to develop such kind of gene therapy because of the cost and because of the regulations. Then another protein is CIS2 that encodes this ZDGSH iron sulfur domain containing protein 2. This is simple protein, single base protein, but it is a more interesting protein because something is known about its function. So it is binding the ferrum sulfur clusters, you remember from the mitochondrial function lecture. It interacts with BCL2, means it could control apoptosis, autophagy, and it is somehow interacting with SERCA, reducing SERCA maybe, and it is necessary for SERCA activity. Most of the disease cases are related to mutations in this wolframin 1 and relatively few less to mutations to this CIS2. In symptoms there is slight difference. The CIS2 deficiency has few more symptoms. And CIS2 is also interesting because it is such kind of anti-aging protein. It has been shown that at old age its expression is decreasing, and if it is activated in mice, then it is, how to say, making the life of old mice better. It's also, if you overexpress it, you activate it, it helps in case of Alzheimer, it helps in case of cardiac problems in mice. So it's such kind of anti-aging protein, and actually people are studying it because of that, not because of its involvement in Wolfram syndrome. So two very different proteins, but they are doing at this, in cell more or less the same things. If you don't have this wolframin 1, then you have ER stress, you have ER calcium disturbances. It's difficult to sell which is coming first. Then you have a mitochondrial dysfunction, then you have a mitophagy and huge effect on cellular bioenergetics. And CIS2 is doing absolutely same things. ER stress, ER calcium problems, mitochondrial dysfunction, mitophagy, and problems in cellular bioenergetics. And they are interacting quite same proteins. So we started to study this actually already 15 years ago, and then we showed, then people were thinking that it is ER protein and it's ER stress disease or ER calcium disease. But the symptoms still are mitochondrial disease symptoms what we have. So then we showed that actually it's the mitochondrial dysfunction that's lead to this disease, that the key factor which is progressing that disease is in mitochondria. So together with us after us several papers came out showing that it's related to ER mitochondrial calcium fluxes and contacts. So but we were still interested because it was not so clear why this ER calcium problems or ER calcium flux to mitochondria, why this happens. And if this happens, then why and how much it really affects the mitochondria. Because a lot of people, even after these papers, they were thinking that mitochondria were secondary layers, just they became affected, but in disease progression they are not so relevant. And then since we are from pharmacology department, we also try to find targets for treatment. So the first question was that why you have less calcium in ER and going to mitochondria? So it is quite tricky to study. So the model we used the primary cortical neurons from 1 day old rat pups. Why you use specifically these neurons? The reason is that they were, in our case, they were, how to say, disease relevant cells. Because if we are looking for the drug targets in case of neurodegenerative neurological disease, then it makes sense to study neurons. And since axons are the most affected part in these cases usually, we focused also to axons only. So we didn't study the cell bodies or somas, but we studied this end of the axons, which is one of the most important places if you have neurodegeneration or neurodevelopmental issues. Then we used the specific plasmids encoding the siRNAs to suppress this wolframin 1 or cis2 in our model in these cells, and then we measured everything related to calcium. ER calcium, cytosolic calcium, or oxoplasmic actually, mitochondrial calcium, ER to mitochondria, calcium flux, and contact sites. So in every different place which was relevant in our context. So you saw this picture or similar picture. So here we have axonal membrane is red, and these green lines, they are ER tubules. So calcium is in these tubules. We used ratiometric dye to have a good normalization, and as you see from this control graph, if we treated the cells with glutamate, this released ER calcium, and we have significantly less calcium in ER, so the dye was working well. Only thing is that to find this specific dye, this 210 means here the calcium, affinity. We had to spend more or less a year to test all different dyes, which will, not dyes, sorry, it's genetically encode sensor, to find a sensor which is perfect in our case. And we did it the same for cytosolic or oxoplasmic calcium. We measured the oxoplasmic calcium. So these measurements alone, they have no value. The value comes only if you quantify these measurements. So here 1 dot means 1 axon. So we picked up the average axons and we measured the calcium level ratiometrically at the end of axon in cytosol or in endoplasmic reticulum. 1 dot, 1 axon. So what you clearly see here is that in groups where we do not have a wolframin 1 or we do not have cis2, we have much less calcium in the endoplasmic reticulum. And in the same groups we have much more calcium in the oxoplasm. These differences were not so visible in cell soma. In soma everything is well balanced, buffered, controlled, and the differences were relatively small or missing even. So what this graph says? This says that we have such kind of situation that we have less calcium in ER, more in cytosol, but nothing more. So this is what is the limitation if you have just this graph. It's just description. It's not showing the reason. So to find the reason why you have this difference, you need to manipulate with system. So what we did, we first expressed the pump, Cerca pump, which pumps calcium from cytosol to ER. what we saw that if we will overexpress this pump, then we have more calcium in ER and less in cytosol. So it's not perfect. You see, this is the wolframine Sirna, and this is the same together with Serca. There is such kind of 15+% effect. The same is in the case of this stool Sirna. So this means that these two things are related, really, that it's not two independent things, that we have less in ER and more in cytosol. No, it's because of, it's not maybe because, but it is at least related to the calcium pumping. So one reason why we have not enough, we may not have enough calcium in the endoplasmic reticulum could be also that it is leaking out. But okay, I forgot this figure. We made one more experiment. We actually overexpressed phospholamban, and this phospholamban is suppressor of Serca. And this alone made situation very similar to what we observed in disease models. So, but okay, so another thing why we don't have enough calcium in reticulum is that it may leak out from reticulum through the release channels. So first we tested this ryanodine receptor calcium channel in ER. So we took the siRNA to suppress this channel, and it was again protective. We suppressed the leak, and calcium normalized in reticulum in cytosol. So until that, everything was logical. We don't have enough calcium in endoplasmic reticulum, it's not pumped in, and it leaks out. Then we went and we tried another channel. Is it also responsible for leak? So we suppressed the IP3 receptor that is in ER membrane, but it was not giving any protection. So there was no improvement. So either it's not involved in leak, or it plays some other roles. So the next question is that, okay, we had more or less the reasons, not enough calcium, but we wanted to see is that the real reason why we don't have enough mitochondrial calcium. So we used similar sensors to measure mitochondrial calcium, ratiometric sensor, and measured it in individual mitochondria. What we saw, it's okay. We saw that we have less calcium in mitochondria, which was expected. But then it was not showing exactly how much this is, again, related to disease and so. So we made such kind of more complicated control experiment. So we took, yes, you can hear now me also. So we suppressed the wolframine 1. As a result, we had less calcium in mitochondria. Then we reintroduced wild type wolframine 1, which is siRNA insensitive, and we restored the mitochondrial calcium levels. But when we used the mutant wolframine 1 plasmids, which expressed disease mutations, then in some cases we got no rescue, and in some cases we got perfect rescue, similar to control. So we had 4 mutations. What is important here, that these 2 mutations which were not working, they were from the patients who have severe, early, relatively strong, early starting Wolfram syndrome. And these 2 were from patients where the symptoms were mild. So we showed that it's indeed disease severity related effect what we have. Okay, so then the next question was that, yes, it was still description. We have less calcium in ER, less calcium in mitochondria, but we don't have a link between these two things. It's not causatively linked. So what we did, we improved calcium level in endoplasmic reticulum. We overexpress CERCA to have more calcium, or we suppress the ryanodine receptor to have a more calcium. And we measured the mitochondrial calcium. Mitochondrial calcium was going back to control level. So this is saying that there is causative relationship. Problems in ER, even corrected, they will lead to mitochondrial improvement. Means that ER is the primary reason. But then we had very strange results with IP3 receptor before, and we decided to check what happens if we increase the IP3 receptor activity. And what was slightly unexpected, that when we increase the IP3 receptor activity, mitochondrial calcium also increased. If we use the dead IP3 receptor, it became even worse. So when we wondered why we have different results, why there is protection, then we were thinking that it is because the IP3 receptor is giving the calcium directly to mitochondria, but ryanodine receptor most likely gives it not to contact site but to cytosolic part. So they are giving calcium to different compartments. So what we concluded then? There is less calcium in ER but more in cytosol or oxoplasm. It can be rescued if we will promote calcium uptake. It could be rescued if we suppress the leak through ryanodine receptor. But strange results with IP3, and we were thinking that it's because of the mitochondria ER contacts. So you saw this figure earlier that we have a sensor. This was originally designed and published by a Japanese research group, which is very good to measure the mitochondria ER contacts. That's the nanolook is the most sensitive, the luminescence. Most sensitive luminescence producer, luciferase assay. So we used that assay, and what we saw, yes, we have less contacts between mitochondria and ER. For some reasons, less. So it's good, but it's again description. Less contacts, but is it relevant? Maybe it is, maybe it's not. So we tried to design control experiments. So one control experiment, what we designed, is that we tried to increase the number of contact sites. We express the GRP75, which is one of the proteins keeping the contact sites together. We overexpressed that. We got more contact sites in control. We got more contact sites in Wolfram syndrome model, as expected. And then we measured mitochondrial calcium. What is good? So you see Wolfram syndrome, mitochondrial calcium less. We increased the number of contact sites. Calcium was coming back to mitochondria. So what is the conclusion is that we don't have enough calcium or enough contact sites for calcium movement from ER to mitochondria. One or both. So then what we also did, we measured the direct calcium flux from the ER to mitochondria. So induced ER calcium release by specific compound and measured how much goes to mitochondria. What we saw is that significantly less calcium is going from ER to mitochondria. Control here, disease, wolframin Sirna here. 3-4 times. And also we saw that with cytosolic calcium. So we really don't have enough calcium in the ER. So we can't release much. If you can't release, or there is nothing to release, then mitochondria will get nothing. So we established what happened from here to here. So we had a problem, not enough mitochondrial calcium. So it's nice, but so what? Is it relevant at all? Is it affecting, how much it is affecting the mitochondrial function? So we measured everything we could. Membrane potential, mitophagy, mitodensity, ATP, NADH. What we saw, the membrane potential. So there are different dyes. This is now dye, not sensors. TMRM, GC1, GC10. So GC10 has its advances because it's ratiometric. If you have a low potential, then it's green. If you have a good potential, then it's red, and the ratio shows how good potential you have. But it has also limitations. It is red, starts to aggregate. If you wait too long, the concentrations are too wrong, then everything aggregates to red. So you should catch the point where everything is still measuring something. And another thing people don't know so well, that it's toxic. In neurons at least, if you use it in concentration that is suggested, then your mitochondria will become round shaped. They are dead, almost dead. So to do this experiment, you should use the lower than suggested concentrations, and you should always check what happened with your mitochondria. Are they still happy? Otherwise, you are having in all groups semi-dead cells. So we did that, and what we saw that we have slightly less membrane potential, or less GC10 red to green signal in disease neurons. Not much. If we treat with uncoupler, then it goes to 0. So some people may say it's not much, but for the membrane potential, it's quite a lot. You can't expect, if you have disease and the carrier of this disease is still living, so it's not so strong disease. You can't expect changes from here to here, because then it's not possible to live anymore. Membrane potential changes in these disease models are always very small. If you use pharmacology, then they are strong, but in the models you can't expect this big change. We have less potential. Then we have mitophagy. So we measured using this schema probe about what I was telling yesterday. We measured that we have much more mitochondria inside of lysosomes if we have a disease models. We counted it. One dot is one cell, means how many dots here we had in one cell, and then this red circle is showing that number. But very big increase in disease models, almost twice more mitochondria inside of mitophagy. Means mitochondria were unhappy. What happens if you have too high mitophagy? it's mainly it's if you have a mitophagy defect, then it's bad because you have accumulation of bad mitochondria. But if your mitophagy is for some reason very strong, then one thing that happens is that you have less mitochondria in cell. It removes so actively the mitochondrial population that you have less mitochondria left. So mitochondrial density is going down in both of these disease cases. It's not only reflecting the mitophagy, but it's mostly, I believe, in our model it reflects mitophagy. And when mitophagy is difficult to measure, you need good microscopes, sensors. Then the mitochondrial density or quantity is easy to measure. Simple microscope image, we use the sensors, but you can use the fluorescent dyes. You will stain, you will count, or you will measure somehow how much you have. Easy to get numbers out. Maybe time consuming, but cheap. Then when we discovered that we don't have enough enough mitochondria, then what is next to measure? No mitochondria, no energy. So we used this Perceval probe to measure ATP at the end of axons. So it's again ratiometric. The red is the axonal membrane. Axonal last segment of axon, and this is the presynaptical terminal, or maybe it's just a growth cone at the moment, which is soon forming the presynaptic terminal. It's a relatively big, good to measure place in axon. So we measured it here. And what we saw there is that we don't have a significant decline in ATP. So this was most, one of the strongest effects we saw. No ATP at the end of axons. When we measured the ATP in soma, the effects were small. Means that the neuron body could be happy, but everything happens at the end of axons and in synapses. It's really necessary to measure what happens there, not what happens in body. The classical ATP measurement is the biochemical assay from all cells. In this case, the biochemical assay shows you absolutely nothing, because problem is in this place. And compared with all signal, this is very small part of signal. So biochemistry will not help, you need imaging. Then what happens in cells when you don't have enough ATP? Because of the mitochondrial suppression. Cell will die, try to do more ATP through the glycolysis. Then you have a lactate accumulation, and you have NADH accumulation in cytosol. So it's another name for that is Warburg effect, which happens in cancers, or people call it reductive stress also. So we use different sensors to measure, do we have more NADH per NAD? And again, we got, in both cases, results showing that there is more NADH in disease groups than in control. So these probes are more tricky. The ATP probe is working always, but with paradox and sonar we have sometimes problems in neurons. These paradox, Perceval, Kaima, calcium sensors, they are all available from that gene. If you need them, you can find them from that gene. The sonar, at least a few years ago, was not there. This we got from the Chinese research group who made it. But then it sometimes takes time to get these probes, because from hygiene it's 1 week you get bacteria, you will grow them up a few more days. So it's 7 days as approximately from order to your plasmid. If you're asking it from another group, then there is paperwork, there are contracts, and it could take months to get it. So the repository, even you pay something there, is always faster. So we made conclusion that because of lower calcium, we have lower membrane potential, lower ATP production, less calcium in axons, who activate glycolysis. We have redox stress because of that. So what remained open here is that how exactly these mitochondrial calcium problems were leading to lower membrane potential. So it was the question, and it was, how to say, we had two independent mechanisms or pathways, but they were not exactly together at that moment. So we were asked to explain that. So what happens, you remember you had pyruvate dehydrogenase in lecture before. So this pyruvate dehydrogenase and actually also other these mitochondrial dehydrogenases, they are calcium sensitive. If you have a calcium, then pyruvate dehydrogenase becomes more active. If you have less calcium, no calcium, then it is passive. So, and this is related to phosphorylation of the pyruvate dehydrogenase. So what we did, we tried to check that if we manipulate with pyruvate dehydrogenase, then what really happens. So how to make pyruvate dehydrogenase more active? You can inhibit it, of course, but in our case we wanted to activate it. And you don't have pharmacological tools or good tools. You can overexpress it, of course, but it's not. It shows that pyruvate dehydrogenase will be relevant, but it's not showing that it is inactive or not enough active. So we overexpress the pyruvate dehydrogenase phosphatase, which removes the phosphor from pyruvate dehydrogenase and makes it more active. So what we got partial rescue. We made the pyruvate dehydrogenase more active using this approach, and this is, you see, the ATP level control, disease, and we got it to somewhere in the middle. When we measured NADH levels, again, control disease, and with phosphatase somewhere in the middle. So it's not strong evidence, not strong enough evidence, but it shows that it plays some role at least. So if it would have been negative, no rescue, then this means that this mechanism has nothing to do with our problem. Then we also wanted to check, is these problems, NADH, etc., ATP, are they related directly to mitochondrial calcium? So we overexpress the miroprotein, which is known earlier by others, by us, to increase the mitochondrial calcium. We increase the mitochondrial calcium, and you see indeed, mitochondrial calcium in control, disease, and we overexpress miro in disease, we got it back. And same with ATP, control, disease, miro, ATP is coming back if we increase the mitochondrial calcium. So we got direct link, mitocalcium, ATP, NADH. Then we did the same at the level of ER to mitochondrial calcium flux. So we overexpressed the IP3 receptor. And the idea was that if we overexpress it, then more calcium is going from ER to mitochondria. So controlled disease, IP3 receptor, half is coming back. Dead receptor, nothing is coming back. So it shows that this ER to mitochondrial calcium flux was partially at least the reason for the ATP loss and so on. And we did the same with Cerca and a few more experiments, and we made conclusion that ER calcium handling defect and ER to mitochondrial calcium flux are the reason for everything that is here. Then again, if you are speaking with medical doctors or clinicians studying that same disease from clinical, perspective, they will say, who cares? So you have in your dish less ATP somewhere, but it's quite far to say that it's relevant. So since it's neurodevelopmental disease, in addition to neurodegenerative, we studied how well the axons are growing in these neurons. So control neurons, axons are growing. If we will knock down the wolframin or cis2, axons are not growing so well. Axons are growing significantly less. So this means that we made a control experiment later also to improve it. And what this means that it's not just some minute effect in our ATP signal, but it has really relevance at the end for the neuronal, not function, but neuronal morphology, which is always affecting its function. Then one of the sub-aims we had there is that we were always studying two proteins, and they always did the same. Both disease-inducing wolframin cis2, when we knocked them down, they always, you remember from the figures, gave us absolutely similar phenotype. How this is, why is the reason? There was one old paper showing that they don't interact. You can't immunoprecipitate them together. But at that time, the antibodies were bad and so on. So we were actually asked to find out how they could do the similar thing, how they are related. So we started from colocalization experiments. You saw this yesterday, that wolframin is in the ER and in axonal terminal, and very similarly cis2 is in the same place. We made colocalization experiments, yes, they were in the same place. Then we also made immunoprecipitations. We started with immunoprecipitations with the artificial constructs. So we overexpressed tagged cis2 and tagged wolframin. We got the interaction, but 2 points. The journal nowadays, they don't accept you overexpress interactions. It's good to have them, to show them, but you should always have also proof that there is endogenous interaction. So we tried to do it. I think we tried 10 different antibodies because mostly they were bad. And then, at least at the end, what we got is that when we immunoprecipitated the cis2 15 kilodaltons, then the wolframin 90 kilodaltons came also with it. It's a tricky to do. You need a good controls. We had here very simple EGG control, but the problem is if you study if you IP cytosolic proteins, then it's easy. But if you IP the proteins which are in cell in the same membrane, in mitomembrane, in EM membrane, then always when you purify your preparation, you will get the membrane fragments at some point which will bring both proteins together to your IP. So you should have a good controls to be sure that this is not that problem. What do you think, is it a proof to show that they interact if we will IP them? It's not, because in this IP mix we have also a lot of other proteins. We IP'd ryanodine receptor, we IP'd SERCA. In one of the paper published after us, they IP'd IP3 receptor. And in all cases, the wolframin is coming together with ryanodine, SERCA, IP3, and cyst is coming. So what we can conclude from this experiment is that maybe they are not directly interacting, but they are in the same complex, and they are in the same complex with main calcium handling proteins, SERCA, ryanodine receptor, IP3. If you want to have data that they really interact with each other, then actually only way is to purify both proteins and to show it. But how to purify exactly wolframin, and so it's not so easy business. We are not strong in biochemistry. We're not doing that, but journal was happy. And okay, they interact with each other indirectly at least, but they interact also functionally. So that if we have here ATP measurement, control cells, wolframin knockdown, less ATP. If we will increase CIS2 expression, we overexpress it, we will get rescue the wolfram deficiency. Or if we have CIS2 deficiency, less ATP, we have a rescue here again. So they are functionally fully interacting at the level of ATP and also at the level of the axonal growth. So axons are not growing, wolfram sirna, they start to grow if we will include CIS2, we will overexpress CIS2. So it's difficult to explain what this means, this interaction. No, really, it's not giving really good conclusions. It just allows to speculate that they are controlling together other proteins. They are controlling maybe SERCA, they are controlling maybe ryanodine receptor, maybe or maybe not, but clearly IP3 receptor. So they are controllers, maybe independently. And what they could sense, speculation again, wolframin could sense ER stress, CIS2 could sense reductive stress because it has these ferrum sulfur complexes. They are maybe sensing this stress, and accordingly they will modulate calcium uptake. This is speculation, of course. And then at the end we had pharmacology. We took all these proteins which we had at the beginning till mitochondrial calcium, and we started to look for the compounds available which are affecting this. So it's SERCA activator, CDN163 is available. Then there is azumolen, which is inhibiting ryanodine receptor. So here we have more calcium uptake. If we inhibit ryanodine receptor, we have less leak. Azumolen is very similar to dantrolen. Dantrolen is the drug used in rare cases to inhibit uranodin receptor. But azumolen is strong, it inhibits it maybe too strongly. Then there is new experimental drug, Rucal S107, that is not inhibiting really fully uranodin receptor, but leak more specifically. It's also in studies, in some kind of, I think it's in clinical studies, in case it was or is, against the post. I don't remember what exactly it was, but it was some neurological disease. Then we took sigma-1 receptor agonist that increases the IP3 receptor-induced calcium flux. And then we used the diabetes drug liraglutide that has very various effects. I mean, it's clear what it is doing, but it's not so clear what it is doing in neurons, why it affects neurons, how much receptors neurons have. And one speculation is that it is increasing the calcium fluxes somehow. And then we had an inhibitor of sodium-calcium exchanger, which pumps or mediates the calcium flux out from mitochondria. So if we inhibit this, then the calcium will remain in mitochondria. So we had altogether 1, 2, 3, 4, 5, 6 compounds. We didn't study this because one group was publishing Science Translational Medicine paper showing that it's protective and also in vivo protective. We continued only with other compounds. So not going to details, we made huge amount of experiments, 2 years for one PhD student. What we got, we measured how these compounds affect, they were all affecting calcium, but how they affect the mitochondrial parameters, mitophagy, ATP, and axonal growth. What we found that they mostly are protective. They mostly give rescue. You see that mostly statistics is quite okay. Only in few cases we don't have statistics. And even in these cases, it is maybe because the drugs were sometimes toxic in some models. We didn't spend much time to find optimal concentration, so we used what was most practical according to literature. Sometimes we had toxicity problems. But in general, all these compounds affecting differently calcium, they were affecting positively the mitochondrial and neuronal health parameters. So the main conclusion then was that for neurons with this disease, the calcium problems are main thing, and that theoretically at least these calcium problems can be corrected. Is it really possible with these compounds, with the exception of liraglutide, which is actually used nowadays off-label in case of Wolfram syndrome? All other compounds are commercially old compounds, commercially available, and you really can't develop them further as a drug. So, but it's still valuable because you have a target. You know that if you have another SERC activator or another uranodin receptor blocker, then maybe it could help. So it's not the drug research, it's more target research. And okay, it's a rare syndrome. It's 1 per 500,000. How many patients? 30,000 patients in the world. But why it's relevant, as you remember, this is the iadomitochondrial calcium contact site disease, and most of our neurodegenerative diseases and also several rare neurodegenerative diseases, they have this MAM component, which is maybe relevant. So that if we have these targets, then we also can test or speculate that if we modulate these, they could in some cases be used in case of other neurodegenerative diseases. So if you want to know more about methods, then it is now published. More or less a year ago it was published. So you can search my name, Wolfram syndrome, you will get it. And editor was quite happy because we had a lot of different components in that paper. First, the paper was not descriptive, but it was showing the cause, that this leads to that, which is good. Another reason was that we had 95% imaging, and it was quantified, it is not images, but everything was quantified. And third, that we did something which is, relevant also, target research. So the editor was happy and included us to this editor highlights, so the last year best papers in the journal. Okay, and we are a relatively small group. We were big 5 years ago, now it's 4 people, 4 PhD level researchers, 4 students, and this specific work was done together with Gert Boltnik, who is from Catholic University at Leuven, from Belgium, who is specialist in calcium. Then with Fingwen Chai, who is from Taipei and who discovered the function of CIS2, and with my postdoc supervisor, who is now retired, with Professor Wexler, who knows everything about the mitochondrial respiratory chain. And also our animal house, head of our animal house, who helped us. We had no in vivo models here, but we used sometimes the neurons coming from wolframin deficient rat to do control experiments. So that is all from me. If you have whatever questions about this presentation, please ask. So it says quite specific, I think, but so it's not relevant to remember what one protein or another protein was doing. It's just good to remember that descriptive science is not anymore popular. You should have cause, you should prove that this is leading to that. Okay.
So this was definitely done for a disease condition, which is extreme case scenario, I would say. But can we use this further in order to, in general, enhance the mitochondrial health?
Mm-hmm. So it's difficult to say, because if we just overexpressed SERCA, or we did all these treatments with our compounds, then we always had a control group where compound or treatment was alone. And it was, in most of the cases, with the exception of when we had toxicity, there was no difference compared with control. So that if mitochondria were okay, or reticulum was okay, then these compounds had no effect alone. So you can't improve happy neurons, happy mitochondria, with these compounds. So one question is that people frequently ask is that this is all cell culture. So where is the animal study? But you can't really do the animal study which includes so many different components. You can't study the mechanism in animal study because animal studies are very complicated. If you want to test something specific, if you want to test is this compound protective, then you will do animal study. You will use different models and zebrafish, mice. You will show treatment is working, etc. But if you have so many compounds, so big pathway, then there is no hope to do in vivo. Of course, it's the best of the best is to do this, to find that, one drug is really good, and then to do with this animal study. But then it takes not, how long it took, 7 years, but then it takes 2 times 7 years. And this is too much because the grant cycle is 5 years, so we can't study, we can't postpone the publication so long. So this is a problem that is nowadays. When I was a young postdoc, then we made circulation research paper, which is more than impact 10, with 3 months, and we had all together there, not sure, 10 or 15 image panels. 3 months, 15 image panels, no really big work. Okay, discovery was nice, but nowadays you want to have a good paper. It's 100 plus figure panels. It's not 3 months. If you are lucky, then it's 3 years. Usually it's more than 5, and PhD students, or even worse, postdocs, they have only 4-year, 2-year periods. They really don't have time for big papers. So therefore we are lucky we have 4 staff researchers who can spend a lot of time to do these things. And PhD students, the first author was PhD student, but for her it took also almost 10 years, but since it was not problem for her because she was she's medical doctor and was working part-time in clinics, and it was more or less hobby for her. So no more, yeah, please. I will come closer, then I will hear you better.
So you mentioned that ATP depletion causes reductive.
Yeah.
Particular threshold of ATP in cell at which point reductive stress turns to oxidative stress.
Okay. So this is a very good question because we can only speculate. The question was how much inhibition in ATP production we need to induce reductive stress or oxidative stress. So, and I can't tell you actually, because it's also a little bit speculation. What happens if you don't have enough ATP? Then glucolysis activates, you have more NADH, but how much you should inhibit mitochondria or ATP to get that, and how strong reductive stress you will get, nobody knows. Even this is speculation that this suppression of ATP is leading to NADH increase, because we really don't have direct evidence to say that they are strongly related. So it's unfortunately it's a good question, but it's bad answer. Okay, then thank you.